Welcome to our video, which will show you how to set up and carry out the GCSE Biology required practical investigating the effect of temperature on the decay of milk. In this practical, we'll be using a colour changing indicator to identify the endpoint of the reaction. For this practical, you'll need some safety glasses, Bunsen burner, 250ml beaker, some 100ml beakers, boiling tubes, a boiling tube rack, a Chinograph pencil or permanent pen, 10ml syringes and a thermometer. You'll also need some cresol red indicator, lipase solution, sodium carbonate solution, a stop clock, and of course we need the milk. Your students first of all put on their safety glasses and then they label one of their boiling tubes as lipase and the other one they're going to label as milk. Your students then use a syringe to add 5ml of the lipase to the tube labelled lipase. Using a clean syringe, your students then need to measure out 5ml of the milk into the tube labelled milk, and to that they add 5 drops of the cresol red indicator. Your students would then add 7ml of the sodium carbonate solution to the milk tube, and that solution should now be a purpley pink colour. Your students then put a thermometer in the milk tube, and they also need to set up a water bath using the 250ml beaker. They then put the milk tube and also the lipase tube into the water bath and they heat it up with the Bunsen burner until it gets to their desired temperature. For example, my first temperature, I'm aiming for 30 degrees using the thermometer in the milk tube. Once the milk's reached the desired temperature, your students then need to move the Bunsen burner away and turn it onto the yellow safety flame. They will then use a drop-in pipette to take 1ml of the lipase solution and place it into the milk and start the stopwatch. They then continuously stir it with the thermometer and wait until there's a colour change. Once the contents of the milk tube have turned yellow, your students would then stop the stopwatch and record the time taken in seconds. Your students would then repeat this procedure with their water bath set at 5 different temperatures. We hope you found our practical advice useful. For expert technical support, please contact the Philip Harris Technical Support Team on techsupport@philipharris.co.uk or visit our blog at philipharris.co.uk/blog.